Hello everyone, this is Professor Beida. We talked about how cellular respiration transforms the food we eat to ATP, but what do plants do? They don't really eat in the normal sense of the word, apart from uh, flytrap, Venus flytraps. As it turns out, plants are getting their energy directly from the source, the sun. Plants use sunlight as well as water and carbon dioxide, which are plentiful in our earthly surroundings, to produce their own nourishments through a cellular process called photosynthesis. This word makes sense because photo means or come from the Greek word for light, and synthesis means to build. So plants use light and small molecules to build bigger molecules that they can then metabolize to produce energy. Other organisms then eat the plant, and other organisms eat those organisms. So plants really are the foundation of the entire food chain. The conversion of or the conversion process that transform uh, the energy of sunlight into chemical energy stored in sugars and other organic molecule is called photosynthesis. Photosynthesis nourishes almost the entire living world directly or indirectly. Uh, and organisms acquire the organic compounds it uses for energy and carbon skeleton by one of two major modes, autotrophic nutrition or heterotrophic nutrition. Autotrophs are self feeders. Auto means self and trophs mean feeders. They sustain themselves without eating anything derived from other living beings. Autotrophs produce their organic molecule from carbon dioxide and other inorganic raw materials obtained from the environment. They are the ultimate source of organic compound for all non-autotrophic organisms, and for this reason, biologists refer to autotrophs as the producers of biospheres. Almost all plants are autotrophs. The only nutrients they require are water and minerals from the soil and carbon dioxide from air. Specifically, uh, plants are photoautotrophs. So plants are photoautotrophs, organisms that use light as a source of energy to synthesize organic substances. Photosynthesis also occurs in algae, certain other protists, and some prokaryotes. Heterotrophs are unable to make their own food. They live on compound produced by other organisms. Hetero mean others. Heterotrophs are the biosphere's consumers. The most obvious other feeding occur when an animal eats plants or other organisms, but heterotrophic nutrition may be more subtle. Some heterotrophs consume the remains of other organisms and organic matter, such as feces, fallen leaves. These types of heterotrophs are known as decomposers. Most fungi and many types of prokaryotes get their nourishment this way. Almost all heterotrophs, including human, are completely dependent, either directly or indirectly, on autotrophs for food and also for oxygen as a byproduct of photosynthesis. The process of photosynthesis most likely originated in a group of bacteria that had enfolded a region of the plasma membrane containing clusters of such molecules. An existing photosynthetic bacteria enfolded photosynthetic membrane functions similarly to the internal membrane of chloroplast. The eukaryotic organelle that absorbs energy from sunlight and uses it to drive the synthesis of organic compounds from carbon dioxide and water. According to what has come to be known as endosymbiont theory, the original chloroplast was a photosynthesis prokaryote uh, that lived inside an ancestor of eukaryotic cells. All green parts of plants, including green stems and ripened fruit, have chloroplasts, but the leaves are the major site of photosynthesis in most plants. There are about 500,000 chloroplasts in a chunk of leaf. Chloroplasts are found mainly in the cells of mesophyll, um, the tissue in the interior of the leaf. This is the mesophyll cells. Those. Uh, Carbon dioxide and enter the leaf, and oxygen exits by way of microscopic pores called stromata. Stromata from Greek word for mouth. Water absorbed by the roots is delivered to leaves in veins, and leaves also use the veins to export sugars to roots and other non-photosynthetic parts of the plant. A typical mesophyll cell contains about 30 to 40 chloroplasts. A chloroplast has two membranes surrounding a dense fluid called the stroma. Suspended within the stroma is a third membrane system made up of sacs called thylakoids, which segregates the stroma from the thylakoid space inside the sacs. In some places, thylakoid sacs are stacked in column called granum. Chlorophyll, the green pigment uh, that gives the leaves their color, reside in the thylakoid membrane of the chloroplast. This is the chlorophyll, reside in the membrane of the thylakoid. The overall photosynthetic equation has been known since 1800s. In the presence of light, the green part of plants produce organic compounds and oxygen and water. We can summarize the complex series of chemical reaction in photosynthesis with this chemical equation. Uh, Water appears on both sides uh, of the equation because 12 molecules of water are consumed and 6 molecules are newly formed during photosynthesis. Um, in 1930s, Van Nel of Stanford University hypothesized that plants split uh, water as a source of electrons from hydrogen atoms, releasing oxygen as a byproduct, and then confirmed by using a heavy oxygen isotope, oxygen 18, as a tracer to follow the path of oxygen atom during photosynthesis. Photosynthesis is not a single process, but two processes, each of which has multiple steps. These two stages of photosynthesis are known as light reaction, the photo part of photosynthesis, and Calvin cycle, the synthesis part. The light reaction are the steps of photo that convert solar energy to chemical energy. Water is split, providing source of electrons and proton, hydrogen ion, and giving off oxygen as a byproduct. Light absorbed by chlorophyll drives a transfer of electron and hydrogen ions from water to an acceptor called an ADP, a nicotinamide adenine dinucleotide phosphate. The electron acceptor, an ADP, is first cousin to NAD, which function as an electron carrier in cell respiration. The two molecules differs only by the presence of phosphate group in an ADPH. The light reaction uses solar energy to reduce an ADP to an ADPH by adding a pair of electrons along with hydrogen ion. The light reactions also generate ATP using chemosmosis to power the addition of phosphate group to ADP, a process called photophosphorylation. Thus, light energy is initially converted to chemical energy in the form of two compounds, NADPH and ATP. The Calvin cycle, named for Melvin Calvin, the cycle begins by incorporating carbon dioxide from the air into organic molecules already present in the chloroplast. In this initial um, incorporation of carbon into organic compound is known as carbon fixation. The Calvin cycle then reduces the fixed carbon to carbohydrate by addition of electrons. The reducing power is provided by NADPH, which acquired its cargo of electrons in the light reaction. Uh, so the figure indicates that the thylakoid of the chloroplast are the site of the light reaction, while the Calvin cycle occurs in the stroma. On the outside of the thylakoid, during the light reaction, molecules of an ADP and ADP pick up electron and phosphate respectively, and NADPH and ATP are then released to the stroma, where they play crucial roles in Calvin cycle. Chloroplasts are chemical factories powered by the sun. Their thylakoids transform light energy into the chemical energy of ATP and NADPH. Light is a form of energy known as electromagnetic energy or electromagnetic radiation. Uh, travels in rhythmic waves. The distance between the crests of electromagnetic wave is called wavelength. Wavelengths range from less than nanometer for gamma rays to more than 1 kilometer for radio waves. This entire range of radiation is known as electromagnetic spectrum. The segment most important to life is the narrow band of from 380 to 740 in wavelength. This radiation is known as visible light because it can be detected as various colors by the human eye. Light behaves as though it consists of discrete particles called photons. Photons are objects, each of them has a fixed quantity of energy. The amount of energy is inversely related to the wavelength of the light. The shorter the wavelength, the greater the energy of each photon of the light. Thus, a photon of violet light packs nearly twice as much energy as a photon of red light. When light meets matter, it may be reflected, transmitted, or absorbed. Substances that absorb visible light are known as pigments. Different pigments absorb light of different wavelengths, and the wavelengths that are absorbed disappear. If a pigment is illuminated with white light, the color we see is the color most reflected or transmitted by the pigment. If a pigment absorbs all wavelengths, it appears black. We see green uh, when uh, when we look at a leaf because chlorophyll absorbs violet, blue, and red light while transmitting and reflecting green color or green light. The ability of pigment to absorb various wavelengths of light can be measured with an instrument uh, called spectrophotometer. This machine directs a beam of light of different wavelengths through a solution of the pigment and measures the fraction of the light transmitted at each wavelength. A graph plotting a pigment's light absorption versus wavelength as called absorption spectrum. Uh, White light is separated into colors wavelengths, those by prism, uh, one by one. The different colors of light are passed through the sample, and we have here chlorophyll in this example. Green light and blue are shown here. We have green light and blue light. The transmitted light strikes a photo electric tube, which convert the light energy to electricity. The electric current is measured by uh, galvanometer. The meter indicates the fraction of light transmitted through the sample, from which we can determine the amount of light absorbed. The absorption spectra of chloroplast pigments provide clues to the relative effectiveness of different wavelengths for driving photosynthesis, since light can perform work in chloroplast only if it is absorbed. Uh, Here shows the absorption spectra of three types of pigments. In chloroplast, we have chlorophyll A, the dark green, uh, the key light capturing pigment that participate directly in light reaction. We have the accessory pigment, chlorophyll B, and separate group of accessory pigments called carotenoids. The spectrum of chlorophyll A and a separate group of accessory pigments called the carotenoids. The spectrum of chlorophyll A suggests that violet, blue, and red light work best for photosynthesis. These two. That means the violet, blue, and red lights work best for photosynthesis since they are absorbed, while green is the least effect, effective color. Uh, this is confirmed by an action spectrum for photosynthesis, which profiles the relative effectiveness of different wavelengths of radiation in driving the process. This graph plots the rate of photosynthesis versus wavelength. The resulting action spectrum resembles the absorption spectrum for chlorophyll A, but doesn't match exactly. This is partly due to the absorption of light by accessory pigments such as chlorophyll B and carotenoids. Exactly happens when chlorophyll and other pigments absorb light? The colors corresponding to the absorbed wavelength disappear from the spectrum of the transmitted or the reflected light, but energy cannot disappear. When a molecule absorbs a photon of light, one of the molecule's electrons is elevated to an orbital where it has more potential energy. When the electron is in its normal orbital, the pigment molecule is said to be in its ground state. Okay? Absorption of a photon boosts an electron to an orbital of higher energy, and the pigment molecule is then said to be is in an excited state. Once absorption of a photon raises an electron to an excited state, the electron cannot stay there long. The excited state, like a high energy state, is unstable. As an excited electrons fall back to the ground state, photons are given off um, and, uh, and after a blue called fluorescence. Okay? An illuminated solution of chlorophyll isolated from chloroplast will fluoresce in the red part of spectrum and also give off heat. Uh, so absorption of a photon causes a transition of the chlorophyll molecules from its ground state to its excited state. The photon boosts an electron to an orbital where it has more potential energy. If the illuminated molecule exists in isolation, its excited electron immediately drops back down to the ground state orbital, and its excess energy is given off as heat and fluorescent light. Uh, a chlorophyll solution here in this flask, excited with ultraviolet light, fluoresces with a red-orange glow. In their native environment of thylakoid membrane, a chlorophyll molecule are organized along with other small organic molecule and proteins in a complex called photosystem. A photosystem is composed of a reaction center uh, complex is surrounded by several light harvesting complexes. The reaction center complex is an organized association of proteins holding a special pair of electron. Um those two special pair of chlorophyll, a molecule and a primary electron acceptor. Each light harvesting complex uh, consists of various pigment molecule, which may include chlorophyll A, chlorophyll B, and multiple carotenoids bound to proteins. Together, these light harvesting complexes act as an antenna for the reaction center complex. When a pigment molecule absorbs a photon, the energy is transferred from pigment molecule to pigment molecule within a light harvesting complex. So, like human wave at sport arena, until it is passed to the pair of chlorophyll molecule in reaction center complex, which boost one of their electrons to a higher energy level and transfer it to a different molecule, the primary electron acceptor, which is molecule capable of accepting electrons and becoming reduced. The solar-powered transfer of an electron from the reaction center chlorophyll A paired to the primary electron acceptor is the first step of light reaction. As soon as the chlorophyll electron is excited to a higher energy level, the primary electron acceptor captures it. This is a redox reaction. Each photosystem reaction center complex surrounded by light harvesting complexes function in the chloroplast as a unit to or it converts light energy to chemical energy, which will ultimately be used for the synthesis of sugar. The thylakoid membrane is populated by two types of photosystem that cooperate in light reaction of photosynthesis. Photosystem 1 and photosystem 2. Photosystem 2 function first in light reactions. The reaction center chlorophyll A of photosystem 2 is known as P680 because this pigment is best at absorbing light, having a wavelength of 680 nanometer in the red part of the spectrum. The chlorophyll A at the reaction center complex of photosystem 1 is called P700 because it most effectively absorbs light of wavelength 700 nanometer in the far red part of the spectrum. However, their association with different proteins in the thylakoid membrane affects the electron distribution in the two pigments and accounts for the slight differences in their light absorbing properties. Light drives the synthesis of ATP and NADPH by energizing the two types of photosystem embedded in the thylakoid membranes of chloroplast. The key to this energy transformation is a flow of electron through the photosystem and other molecular components built into the thylakoid membrane. This is called linear electron flow, and it occurs during the light reaction of photosynthesis. Um, There are 8 steps in linear electron flow. A photon of light strikes one of the pigment molecule in a light harvesting complex of photosystem 2, boosting one of its electrons to a higher energy level. As this electron falls back to its ground state, an electron in a nearby pigment molecule is simultaneously raised to an excited state. The process continues with the energy being relayed to other pigment molecule until it reaches the P680 pair of chlorophyll A molecule in the photosystem 2 reaction center complex. It excites an electron in the pair of chlorophylls to a higher energy state. And then an excited electron from P680 is transferred to the primary electron acceptor. We now call it P80 plus. And then here in step 3, H2O, water is split by enzyme. And the electrons are transferred from the hydrogen atoms to P680 plus, thus reducing it to P680. In step 4, each electron falls down an electron transport chain from the primary electron acceptor uh, photosystem 2 to photosystem 1. And then energy released by the fall drives the creation of proton gradient across the thylakoid membrane. Diffusion of hydrogen ion or protons across the membrane drives ATP synthesis. Okay, and here in photosystem 1, like photosystem 2, a transfer light energy excites P700 with which loses an electron to an electron acceptor. Each electron falls down an electron transport chain from primary electron acceptor photosystem 1 to protein pyridoxine, and then the electrons are then transferred to an ADP from protein pyridoxine to reduce it and produce NADPH. We produce NADPH. The energy changes of electrons during their linear flow through the light reactions are shown in a mechanical analogy here. Although the scheme shown here may seem complicated, don't lose track of the big picture. The light reactions use solar power to generate ATP and NADPH, which provide chemical energy and reducing power, respectively, to the carbohydrate synthesizing reaction of the Calvin cycle. The certain cases, photo-excited electron can take an alternative path called cyclic electron flow, which uses photosystem 1 but not photosystem 2. The cyclic flow is a short circuit. The electrons cycle back from pyridoxine to the cytochrome complex, then via plastocyanine molecule to a photosystem P700 chlorophyll in the photosystem photosystem 1 reaction center complex. There is no production of an ADPH and no release of oxygen that results from this process. On the other hand, a cyclic flow does generate ATP, but not an ADPH. Photo-excited electrons from photosystem 1 are occasionally shunted back from pyridoxine to chlorophyll via the cytochrome complex and plastocyanine PC. This electron shunt supplement the supply of ATP via chemosmosis, but produce no NADPH. The shadow of linear electron flow is included in the diagram for comparison with the cyclic route. The two pyridoxine molecules in the diagram are actually one and the same. Okay? The final electron carrier in the electron transport chain of photosystem 1, although it is duplicated twice to clearly show its role in two parts of the process. Uh, Chloroplasts and the mitochondria generate ATP by the same basic mechanism. An electron transport chain pumps protons across a membrane as electrons are passed through a series of carriers that have progressively more affinity for electrons. Thus, electron transport chain transform redox energy to a proton motive force. Potential energy stored in the form of an hydrogen ion gradient across a membrane. An ATP synthase complex in the same membrane couples the diffusion of hydrogen ions down their gradient to the phosphorylation of ADP, forming ATP. Some of the electron carriers, including the iron containing proteins called cytochromes, are very similar in mitochondria and chloroplasts. The ATP synthase complexes of the two organelles are also quite similar, but there are uh, noteworthy differences between phosphorylation in chloroplast and oxidative phosphorylation in mitochondria. Both work by way of chemosmosis, but in chloroplast, the high energy electrons drop down the transport chain, come from water, while in mitochondria they are extracted from organic molecules which are thus oxidized. Chloroplasts don't need molecules from food to make ATP. Their photosystem capture light energy and use it to drive the electrons from water to the top of the transport chain. In other words, mitochondria use chemosmosis to transfer chemical energy from food molecules to ATP, whereas chloroplasts use it to transform light energy into chemical energy in ATP. The spatial organization of chemosmosis differs slightly between chloroplast and mitochondria. In both kinds of organelles, uh, electron chains pump proton across a membrane from a region of low concentration, light gray in the diagram, to one of high concentration, this dark gray. The protons then diffuse back across the membrane through ATP synthase, driving the synthesis of ATP. Electron transport chain proteins in the inner membrane of the mitochondria pump protons from the mitochondrial matrix to out to the intermembrane space, which then serve as a reservoir of hydrogen ion. Similarly, electron transport chain proteins in thylakoid membrane of the chloroplast pump proton from stroma uh, into the thylakoid space, from stroma to the thylakoid space in this side, or I'm talking about chloroplast, which function as a hydrogen ion reservoir. In mitochondrial, protons diffuses down their concentration gradient from intermembrane space through ATP synthase to the matrix, driving ATP synthesis. In the chloroplast, ATP is synthesized as the hydrogen ion diffused from the thylakoid space back to the stroma through ATP synthase complexes, whose catalytic knob are in the stroma side of the membrane. Thus, ATP forms in the stroma, where it is used to help drive sugar synthesis during the Calvin cycle. Energy from light um, excites an electron from chlorophyll, which is transferred to the primary electron acceptor in photosystem 2. Uh, in the thylakoid space, water is split into two electrons and which replace chlorophyll's electron twice, and uh, two hydrogen ion, which contribute to the high concentration in the thylakoid space, and oxygen atoms, which joins another oxygen, forming oxygen gas. Cold arrows track the flow of electrons. As electrons travel down the electron transport chain, four hydrogen ions are pumped into thylakoid space, increasing hydrogen concentration and decreasing the pH. In chemosmosis, uh, the diffusion of hydrogen ion from the thylakoid to the thylakoid space back to the stroma uh, down the gradient powers ATP synthase and produce ATP. Low energy from water and end up in NADPH formation, removes hydrogen from stroma and decreasing its hydrogen ion concentration and increasing its pH. The Calvin cycle takes place in the stroma. It's similar to the citric acid cycle in that starting material and is regenerated after some molecules enter and other exit the cycle. However, the citric acid cycle is catabolic, oxidizing acetyl-CoA and using the energy to synthesize ATP, while Calvin cycle is anabolic, building carbohydrate uh, from smaller molecules and consuming energy. Carbon enters the Calvin cycle in the form of uh, carbon dioxide and leaves in the form of sugar. The cycle spends ATP as an energy source and consumes NADPH as reducing power for adding high energy electrons to make the sugar. The carbohydrate produced directly from Calvin cycle is not glucose. It is actually a 3-carbon sugar called glyceraldehyde 3-phosphate. For the net synthesis of one molecule of the glyceraldehyde 3-phosphate, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide, one per turn of the cycle. The Calvin cycle has 3 phases: carbon fixation and reduction, and the third one, regeneration of carbon dioxide acceptor. The Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. The Calvin cycle is anabolic. It builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH. Carbon enters the cycle um, as carbon dioxide and exits as sugar. For net synthesis of one glyceraldehyde 3-phosphate, the cycle must take places 3 times, fixing 3 molecules of carbon dioxide, one of each turn of cycle. The Calvin cycle has 3 phases, as I said before: carbon fixation and reduction and regeneration of ribose sugar. In carbon fixation phase, the binding of carbon dioxide to a 5-carbon sugar named ribose and ribose 5-phosphate is catalyzed by ribose biphosphate carboxylase oxygenase or Ribose, and the 6-carbon intermediate molecules uh, is immediately split into 2 molecules of 3-phosphoglycerate for each carbon dioxide fixed. In phase 2 reduction, each molecule of 3-phosphoglycerate is altered through phosphorylation by 6-ATP. Here 6-ATP were consumed and turns to ADP because their phosphate group is added to 3-phosphoglycerate to form 1,3-biphosphoglycerate. For every 3 carbon dioxide molecules that enter the cycle, 6 molecules of glyceraldehyde 3-phosphate are formed. Only one of these can be counted as a net gain of carbohydrate. In the third phase, regeneration of ribose biphosphate, the remaining 5 molecules of glyceraldehyde 3-phosphate are rearranged in a complex series of reactions, yielding the 3 molecules of ribose biphosphate. 3 additional molecules of ATP are used to facilitate the regeneration of ribose biphosphate. So again, 3 molecules of carbon dioxide enter one at a time. In the first phase, carbon fixation, Ribose becomes a short-lived carbon intermediate with phosphates. It then becomes 3-phosphoglycerate. 6 ATP enter and 6 ADP exit. 1,3-biphosphoglycerate is formed. 6 NADPH entered and 6 NAD exit, plus another 6 phosphate group or ion exit. Phase 2 reduction, 6 glyceraldehyde 3-phosphate, 6 molecules of glyceraldehyde 3-phosphate are formed. They have an output of glyceraldehyde phosphate, a sugar with a glucose and other organic compound within the cycle. 5 glyceraldehyde 3-phosphate continue and then enter the regeneration of ribose biphosphate. The carbon dioxide acceptor, this one we call this carbon dioxide acceptor molecule, ribose biphosphates are formed. These continue toward the original input of three molecules of carbon dioxide from Ribose. Uh, the energy entering chloroplast as sunlight gets stored as chemical energy in organic compounds, and sugars made in the chloroplast supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. Plants store excess sugar as starch in structures such as roots, tubers, seeds, and fruits. In addition to food production, photosynthesis produces the oxygen in our atmosphere.